How is it that we find ourselves surrounded by such complexity, such elegance?
The genes of you and me, the genes of you and me, we're all made of DNA. We're all made of the same chemical DNA. We're all made of DNA.
Hi, you're listening to DNA Today, a podcast and radio show where we discover new advances in the world of genetics. From genetic technology like CRISPR to rare diseases to new research, we have you covered. For a decade, DNA Today has brought you the voices of leaders in genetics. I'm Keira Deneen. I'm a certified genetic counselor and your host. My guest today is Dr. Stephanie Ryan. Dr. Ryan is a chemist, parent, and social media influencer who uses her background to create educational products and content. Since DNA Day is coming up on April 25th, Dr. Ryan's going to share an easy DNA experiment that you can do at home with kids or in the classroom. So Dr. Ryan, thanks so much for joining us today, and we're going to get crafty.
Yeah, I'm really excited to share. And you know what? Adults will like this too. It blew my husband's mind that we could do this.
I have to say, I loved doing these experiments. Like even in college, sometimes we would do certain experiments, and it would be like 8th graders were visiting for a tour, and I'm like, but we get to do the experiment today. Like I'm sometimes more excited than the 8th graders. So in this genetics experiment, what are we trying to do? What's hopefully our outcome?
So there are two outcomes. One is we're going to extract DNA from a strawberry using things in your kitchen and maybe your medicine cabinet. And the second is to show students that science can happen everywhere and it doesn't need a fancy piece of equipment like they saw on CSI or things like that to get the DNA, that we could see the DNA in the strawberry.
Yeah, I think that's very well said, that a lot of people think, oh, if we're doing an experiment, we need to order from some laboratory and have very specific high technology. But honestly, a lot of this is probably already at your house. So what are some of the components that we need, some of the materials that we'll need to conduct our experiment?
Okay, so you're going to need some table salt. You will need some dishwashing liquid. You will need some water, 2 plastic cups, a couple of fresh strawberries with the green part cut off, and you need isopropyl alcohol that has been put in the refrigerator for a while. So that's a safety thing. Be careful you're putting it where no one thinks it's a drink, but it needs to be cold. And before you ask, why does it need to be cold? I actually don't know the answer to that. And that's something I'm going to look up after the show with my son when we do this, because I love to share with him that I don't know all the answers. And then you need a coffee filter.
So
and a
most
baggie.
of that, yes. So I would say most of that, like I'm checking off. I'm like, I have this at my house, this at my house, this at my house. So that's very helpful. So as we're starting this experiment, what are we doing once we've gathered all of our materials? Like, let's go through the process together.
Definitely. I like to say kitchen chemistry is kind of more art than science. You can just kind of use what you want. I believe it was 1 teaspoon of salt into one of the plastic cups. And then I've got half a cup of water. I'm going to add to that.
And then if you guys are watching on YouTube, you can actually see her doing this. So switch over to YouTube if you're listening to this as an audio podcast.
And then you're going to add some soap, the liquid dish soap. I'm just doing a couple squirts of it.
Yep, you're just eyeballing it like crafty. Yep. Love it.
I'm a mom at home, and that's the best I can do because that's what my toddler would do. So
exactly. They're not going to perfectly measure.
Exactly. So you've got this ready. This is what we're going to use to lyse the cells in the DNA. This is going to break that out so the DNA comes out. So we're going to hold on to that for a minute. And now you're going to take
what's cool about that is, so basically the liquid detergent, and correct me if I'm wrong, because you've probably better read on this, more recently read than I am, but that is what's breaking up the cell membrane, because on our cell membrane, we basically have 2 layers of fat, like fat cells, and part of a fat cell, one part is hydrophobic and one part is hydrophilic. So when we're talking about the hydrophobic part, that's going to be the inside, and then the hydrophilic part is on the outside. But with the liquid detergent, that's what's breaking that down so that the cells are popping open. Am I getting that right?
Yep. And the
right,
DNA just like
right.
shoots out. So what I've done is I did a cooking show style. This was 3 strawberries. I cut the tops off because you don't want the leaves in there. And I just sat here while we've been talking and squished them up pretty well. And now what I'm going to do is add some of that stuff we just made into this baggie to help break those cells up. Now the key here,
all together with the mashed up strawberry.
Yeah. So I'm going to add a couple teaspoons of it. Again, art, not science.
Art and science, a lovely marriage.
Yes. Okay, so what you're going to do then is make sure you zipper the bag because this is really messy at this point. What you want to do is gently mix because you don't want to get all those bubbles from the soap because that could make it harder to strain in a minute. So
you're gently massaging the bag there.
Yep, just a little bit. This is the first time I've used palm olive instead of Dawn, so we'll see if different soaps
see if it's better or worse.
Make difference. Yeah.
I'm sure this will be a good thing for kids to do. Be gentle, because kids are gonna want to squeeze it really hard. But
definitely.
Yeah, good fine motor skills right there.
All right, so now what you're gonna do? I'm gonna keep doing this for a minute while we're talking.
Yeah.
What we're gonna do is take the coffee filter and place it inside another cup. And then what we're going to do in a minute is we're going to pour this in and strain out the pulp. So just like you would do with juice, if you're not a pulp person, that's all you're doing. I imagine you could use a paper towel too. I actually had trouble finding a coffee filter today, so that was going to be my next step. So a lot of these things you can use something else.
Yeah,
okay.
If you're a Keurig house, you might need to grab a paper towel instead of a coffee filter.
Yeah. So I am not going to hold this up while I'm doing it because I don't want to spill it all over my office because I feel like that would be a total
and
disaster.
The precious blue Yeti mic there.
All right, so I'm going to pour it in and then I'll hold it up. And the more strawberries you use, the more you're going to have here. But you really don't need a lot. Like you could use 1 strawberry. Let's say that's all you had at home today because 1 produces quite a bit of liquid.
Okay. How many did you use for the amount that we saw there? Like 3?
I used 3. Okay. And so far I only poured part of it in so you can see how it strains.
Okay.
So what you're getting...
fluid.
Yeah, and so this fluid actually has some DNA in it. You can't see it yet, but it's there. So strain. I don't think I'm going to do the rest, just because that will take forever to do all of it. What I will do now is wipe my hands because I have now made a mess. So the isopropyl alcohol had to be refrigerated. And again, maybe by the time this gets put up, I'll know the answer to why it had to be cold. But it does have to be cold.
Right, we could do an update in the blog post here.
It's something to do with making the DNA precipitate out, but I'm not sure why it has to be cold to do that. And maybe it has to do with being miscible with the water. That's the only thing I can think of. So
I know. I'm trying to think too. I'm like, why would that be the case? But it's important for some reason.
That's
homework for you guys too.
So I don't have a lot in the bottom right here, but like I said, I didn't use everything I did.
Right.
And you're
Looks
going
like
to
you have about an ounce, I'd say.
Yeah, you're going to add this approximately, again, the same amount of isopropyl alcohol into it.
So 1-to-1 ratio we're aiming for.
And if it's 1.2, whatever,
yeah.
Again, science at home. What you're going to do is you're going to gently stir it, and then you take a stick. I forgot to put this in the materials list. It's like a skewer, or you could use a toothpick. Okay. You could use a pencil. Or a chopstick. You could use any of these things. Yep. And you gently swirl it, and you see...
Oh, would you look at that?
We have DNA.
Wow. So in the 8 minutes that we've been recording, you just isolated DNA from strawberries.
Yes. And the cool thing is, I don't know the answer to which berries produce more. And that's something you could do with your kids because you just made a bunch of this solution that you could just keep trying with other fruits, berries. I've heard people try it with meat before. Just go through and see, do frozen strawberries produce more than fresh strawberries and things like that? You can do these experiments with your kids, and that could be a fun setting up their own experiment activity.
Yeah, and great for them to see, like, in terms of experiments in the real world, in terms of, when we're like, we don't know the answer when we're doing this experiment. Cool to see that that's how things are done. Like, okay, we're going to compare. So that would be really interesting. If you guys try that out, tweet, post on social media, tag us to say, like, okay, what worked better? What did you try? Blueberries, strawberries, bananas? Did you try meat? Frozen, like you said, what worked out better? But it's cool that you can see it's very, like, from my perspective, it looks like it's sticky. It almost looks like clear snot.
Yep, that's what my son says. It is snot. That's the 5-year-old answer to that.
Yes, apparently that's where my brain is today. But yeah, very fun, very easy to do the experiment. If you enjoyed DNA Today, especially episode 139 with Dani Shapiro, who discovered she was donor conceived, then you'll also enjoy more shocking discoveries from DNA testing and a new podcast I love, Biohacked Family Secrets. It's a brand new podcast all about hidden ancestry, medicine, and technology. The show follows the children of anonymous sperm and egg donors, some with dozens or even hundreds of half-siblings, who accidentally learn the truth because of commercial DNA tests like 23andMe. The podcast drops some bombshells and shows how new technology is unraveling the deep, dark secrets of the baby business. Along with scandalous personal stories, Biohacked Family Secrets also reveals unexpected ethical, scientific, and political questions. It's a vital listen for anyone who loves wild stories about science, tech, and history, or even just messy family drama. You can find Biohacked Family Secrets wherever you're listening right now. New episodes drop every Wednesday. PerkinElmer Genomics is a global leader in genetic testing, focusing on rare diseases, inherited disorders, newborn screening, and hereditary cancer. Testing services support the full continuum of care from preconception and prenatal to neonatal, pediatric, and adult. Testing options include sequencing for targeted genes, multiple genes, the whole exome or genome, and copy number variations. Using a simple saliva or blood sample, PerkinElmer Genomics answers complex genetic questions that can proactively inform patient care and end the diagnostic odyssey for families. Learn more at PerkinElmerGenomics.com. Are you seeking a career with purpose in the healthcare space? I'd recommend checking out Worldwide Clinical Trials, a contract research organization that partners with pharmaceutical and biotechnology companies to advance development of new medicines. Apply your passion at Worldwide, where they take an entrepreneurial approach backed by scientific expertise to help bring treatment options to patients. With a global team of 3,000 experts, you'll be inspired by bright thinkers, dreamers, and doers who innovate and invent for a brighter tomorrow. Perks include unlimited flexible paid time off, limited travel requirements, and the ability to work remotely. Bring your talents and passion and join the team at worldwide.com. Are you seeking a position as a genetic assistant? People in these vital roles aid with clinical and research patient communication, data entry, genetic testing coordination, and administrative tasks. Therefore, training is key. We recommend the Genetic Assistant Online Training Program at Johns Hopkins University School of Medicine. This online program provides knowledge and skills to learners considering genetic assistant positions or those recently hired into these roles who need job training. The program consists of 2 10-week instructor-led courses. All you need is basic understanding of science, particularly biology. At the successful completion of the program, learners will receive a certificate of completion from the Johns Hopkins School of Medicine and the McKusick-Nathans Department of Genetic Medicine. Applications are closing for the summer cohort, which starts June 6th, and the fall cohort starts September 12th. A limited number of partial tuition waivers are now available to offset the cost of the program. Don't wait. Check out the Genetic Assistant Online Training Program at Johns Hopkins University School of Medicine now. Click the link in the show notes for this episode for more information. You can also access that link via the blog post for this episode, available at dnapodcast.com. What are some good questions to ask kids while you're doing the experiment to really get their science mind going?
I would say, what do you think is happening? Like, how did this come out? Which substance here do you think did it? Kind of stepping through in the same way we were doing. Wonder what the soap does. And you know what? Your 5-year-old's not going to know the answer to that, and it is totally fine. They might say, it cleans the strawberry off. And you're like, cool, that works. That works for me. Let's see what happens, And really at this point, it's about them making an observation and doing the experiment themselves. And then knowing that DNA is inside a strawberry, to start putting those pieces together. Now, the older they get, you could talk about lysing of the cells and all of that stuff, and then setting up a proper experiment. Like, if you wanted to set up an experiment, what would you need to keep as a control variable? What are we changing? What is our measured variable? Like that. So those kinds of questions. But mostly when I work with my son, who's 5 now, but was 3 when we started all of this, is what has changed? What has stayed the same? Can we ever get the strawberry back from this? No, this was kind of a technically chemical reaction because it broke the membrane, but it released the DNA intact. So that's tricky to ask. But no, the DNA can never go back in the cell again. It's done so.
And I think another question, as kids get older, something that always intrigued me, like maybe 8th grade and beyond, was like how many chromosomes different fruit have, and then compared to us. So I think as, obviously for Charlie's your son's name, right? So for
yes.
Charlie, that's a little much to be like, how many chromosomes does a strawberry have? But, when you go back to that when he's 13 or something, that might be, interesting conversation to have there.
Definitely. And if you're doing other fruits, something to get into is seedless fruits and how they actually can't reproduce because they don't have seeds. And how did they become this way? And you could see different watermelon varietals have different numbers of chromosomes, and the one that comes out seedless, and looking at that, you can take it a step further and have them test it out themselves and kind of get more into that. And there are gel electrophoresis kits and stuff like that now that you can do at home. So if your kid really is into this, let them go gung-ho into it.
Yeah, I definitely remember the first time I did a gel electrophoresis and just how cool it is. And my high school science teacher, shout out Dr. Kessler, one of the things we did in our classroom, just as a visual aid for how gel electrophoresis works, is we had all of our desks. Let's say there was 20 in the room, right? And they all were in rows, perfect columns. She's like, all right, everybody go to the back of the classroom. So we all went to the back, and she messed up some of the desks. So it was like an obstacle to get forward, but some of them were still a clear shot, like an aisle to the front of the room. She said, okay, now everybody run forward for 5 seconds, stop. And some people were able to get further because there wasn't things in the way. And so I think that was just oh, that's how gel electrophoresis works. The DNA is able to make it through somewhat. And if they're smaller, so some of us were on our own, some of us had our hands holding another person. So it's we're a bigger chain of DNA. And I think that was just cool and nice to be able to visualize that. And a fun activity, as us running around desks and everything in the room. But I think ways that we can make DNA, genetics, and science fun and interactive. I remember that was 10 years ago that I was in high school. So I still remember doing that experiment, So I think that's just so great to have kids have hands-on things, and they'll remember that.
Yeah, and they're hands-on in a way that isn't just surface level hands-on. Oh, here, we're going to do an activity where you move around, and it simplifies it too much that it doesn't actually pertain to what you're doing. That is a great way to do that, of where you were holding onto somebody's hand, dragging them along. You're a larger piece of the protein that is taking longer. But no, I totally agree. I loved gel electrophoresis because it was like a puzzle. When you would use it to which protein could this have been, or where did this get lysed? And it was whew, that's, and it's you're working through, and it was almost like doing a Sudoku.
Yes. Yeah. I love those too. So let's say that while a parent, caregiver, teacher is doing the experiment with kids, and a kid answers a question or says something that's incorrect, how do we handle that? Because obviously we want to foster that scientific mind, but we also don't want them necessarily to go forward thinking something incorrectly. I mean, how do you handle that with Charlie? If he says something that's incorrect that that you're oh, that's not how it works.
I bite my tongue and I let it go. Because it is more important that he is building his own mental model. And whether that mental model is correct or not, he needs to build it. feel confident in his own voice and showing what he thinks is happening. And then what I do, takes a lot of extra work as a parent, is you find something that helps shake up that mental model. So I find a way to like really highlight something that's wrong, like that I would have corrected him on, and be like, okay, now try this. And then it won't work. That model will not work in that way. And he'll go, oh, and then he has to think something new. And so it takes more effort on our part. But my thoughts are, especially with the young ones, it gets fixed in the end. Just keep giving them these opportunities, because if you shut them down and go, no, like that, then they aren't going to speak up. They're not going to be as excited to share what they're thinking. But as a scientist and an educator, that one is the hardest for me as a parent.
Yeah, because right away you want to be like, no, it's actually this. And you're like, oh, hold on. But I love what you said for Charlie. If he says something that's incorrect or kind of on the wrong track, it's like, show him something that he's gonna question what he just said and understand where it's going, so that he's discovering it on his own. So he's working through that, because someone just telling you how it works, you know, as we've been talking about, like hands-on, you realizing yourself, that's what you're gonna remember. Just like when you make your own mistakes, that's what you pull from.
Definitely. Like the other day we were in the car and we were talking about, I forget, there was something he got to do that was fun. And he was like, well, I'd like to read first because I need to get energy. And I thought to myself, wow, you are an introvert, really? And he was like, no, mommy, reading gives me more red blood cells, which increase my energy. And the way he said it was like, duh.
This 5-year-old talking about red blood cells. Oh my God, I love it.
He was just like, duh, mommy, you know, kind of like that. And I was like, that makes no sense. But I'm like, okay, like, if that's what you think it does, I'm gonna let you think that. It's super cool that you're talking about red blood cells and you know that there's something in your body that does something, you know? So we'll get there.
If he likes reading, keep that going, right?
Yeah.
Like, that's gonna serve him very well in life. So how do you approach something if Charlie asks you a question and, I mean, this probably doesn't happen too much, but you just don't know the answer?
It happens all the time.
It happens all the time. I'm like, this woman has a PhD. Like, she's got most of the answers. But I mean, yeah, sometimes they ask us like the philosophical questions or whatever. How do you approach that with him?
I Google it. I learned side by side with him, which I think is super important for parents to do, because I know when I was a kid, your teacher was just right. Your parents were just right. They didn't have time to go to the encyclopedia, look something up and show you. So it was just like, okay, the adults are right. And I think that it's important for kids to see that everybody needs other people's answers sometimes. And we're not experts at everything. And we're going to learn our whole life. Learning doesn't end in 12th grade. And so showing him that, look, I'm learning right next to you. I have no idea. And this is how I approach it. And even when I'm writing things in my daily job, like I still look things up all the time. I contact experts. I'm not an expert in everything. And so like we were saying with the isopropyl alcohol, why does that have to be cold? I'm actually not sure. But that's exciting because it's like, look, you don't know. I don't know. Let's figure it out. And so the figuring out process becomes kind of a bonding moment a little bit with your kid, but also a good moment for them to see modeled behavior.
Yeah, when you don't know something, let's research it. How are we going to research it? Because it's like teach a man to fish, right? So you're setting them up to know how to find answers in the future and not just like supplying them with one in the moment. And more exciting. You're like, it's an adventure. Like, I don't know, let's find out together. Yeah. I think it brings that excitement to science. And I think there is such an excitement behind we don't know. And it's like, how are we going to find out? Or like, when are we going to find out if us as a human race doesn't have the answer yet? So I love that approach and just being human because, yeah, I agree with you. Like growing up, it was like, you know, oh, mom knows everything. And then you get to a certain age and you're like, oh, hold on, mom doesn't know everything, you know?
Yeah, and it will eliminate those kinds of behaviors where people just kind of make up an answer so that it sounds like they know what they're talking about, and that's the worst. And so, like, I don't want him to do that. So I'm trying to make sure that that's not something he does. It's okay to say, I don't know.
Yeah, totally agree with that. And as I've said, you've earned your PhD. It was in learning sciences with a focus on chemistry education, which you know makes sense with everything we've been talking about. Do you have any other advice for making science understandable for kids? Is there any other certain approaches you take with Charlie or anything like in your day-to-day job?
Yes. So one of the things that I do, and it takes a while. It's like putting on a pair of glasses that like really messes things up for a bit, but then you get it in focus and you can figure it out, is to break it down and try to visualize it through their eyes. So sometimes I'll do something with baking soda and vinegar, and to me that is so boring at this point. I've done this 150 times during the pandemic. I don't want to do it anymore. And I'm like, oh, it's going to be a waste of time. And he's just like, wow, the bubbles have a gas in them. And I'm just like, okay, all right, and so pulling it through their eyes, and then not reinventing the wheel. So they don't need to look at it too in depth. You can talk about solids, liquids, and gases, things that have changed versus stayed the same. Just at that level. And that's all they really need, is to like learn how to make observations, make comparisons. And it doesn't have to have fancy chemicals. It can be things in your kitchen. And it's, it just take a step back. It's not in the lab. And if you had a bad experience in chemistry or biology in high school, which I know many parents did, you actually know a lot more about the world around you than you think you know, because you're looking at it in this different way. And that's not the way we're talking about it. We are talking about it in terms of help your kid explain the world around them, not regurgitate the periodic table.
Right. Learn through exploring and explaining like what you see. It doesn't need to be like as official as, we had in sitting in high school classrooms and everything. The Big Biology Podcast dives deep on some of the most provocative and exciting topics in biology today in a fun and accessible way. In each episode, hosts Art Woods and Marty Martin, biology professors themselves, talk to leading scientists and journalists from around the world about the biggest, most cutting-edge topics in biology. From human consciousness, human origins, new directions in evolutionary theory, to the emergence and spread of zoonotic diseases like COVID-19 and much more. Whether you're a researcher, teacher, student, or simply curious about science and biology, you'll love the entertaining and deep conversations about biology and the exposures to the research they bring. Learn more at bigbiology.org and subscribe, rate, and review on Spotify, Apple Podcasts, Google Podcasts, or wherever you get your podcasts. Are you interested in the rapidly growing field of genetics and want to learn more about clinical genetics, molecular genetics, and laboratory science? Then you should check out the Genetic Assistant online training program at Johns Hopkins University School of Medicine. By taking part in the program, you'll be joining both national and international learners with the same passion for genetics. Interact directly with your Johns Hopkins instructors and fellow learners throughout the program. Applications are closing for the summer cohort, and there are spots available for fall 2022. For more information, use the link in our show notes. If you're listening to this through a podcast app, you can also access the link at dnapodcast.com. Did you know PerkinElmer Genomics was one of the first laboratories to offer whole genome sequencing on a clinical basis? Whole genome sequencing can maximize clinical diagnostic yield for patients. With turnaround time of 4 weeks for the proband sample, PerkinElmer's whole genome sequencing test is designed to provide access to additional valuable information compared to an exome. PerkinElmer also offers prenatal whole genome sequencing as well as ultra-rapid whole genome sequencing for critically ill newborns using dried blood spots. The ultra-rapid genome has a turnaround time of 5 days and includes mito, chromosomal CNV analysis, STR-TNR screening, and biochemical analysis. Also, listen back to episode 176 with Dr. Madhuri Hegde, where we explored the power of whole genome sequencing, which also happens to be one of my favorite episodes of DNA Today. And stay tuned for a couple more episodes with PerkinElmer soon. Discover all that PerkinElmer Genomics has to offer at PerkinElmerGenomics.com. Are you developing a new therapeutic? Need a partner to help? No matter where you are in your development program, Worldwide Clinical Trials is available to guide you. With more than 35 years of global experience founded on medical science, Worldwide will execute your trial with confidence. Worldwide Clinical Trials has an extraordinary depth of medical expertise in key therapeutic areas, including cardiovascular, metabolic, neuroscience, oncology, and rare diseases. Unique logistical challenges and lack of precedents can make rare disease trials intimidating. Worldwide offers customized insight, deep expertise, and novel solutions to get the most out of your efforts. Worldwide uses their extensive network of global resources to meet or exceed patient recruitment and retention goals. This is achieved by combining the benefits of global scale with personal service and senior management accessibility. Stay tuned for our full episode interview with Worldwide here on DNA Today. Learn more about how Worldwide can be your clinical trial partner at worldwide.com. You, I think, have another home experiment that you do in terms of, being in this genetics area with DNA Day right around the corner. What do you have up your sleeve there?
So I'm a chemist, but I love genetics. And you might think I'm actually a geneticist by how much I talk about genetics versus chemistry.
I was going to say, we are very on the same page. It's just like geeking out over genetics. So
it is my second love, I guess. Maybe my first.
We won't tell anyone. Just secret between us and the listeners.
So I have this activity in terms of puzzle pieces. So base pairs, that's what they are. And that's all we need them to think about. Don't think about actual structure. And I just did a random cutout of what goes together. And so I did it for RNA. I did it for the same shape for DNA so that we can do that.
If we're pairing adenine, we can pair it with uracil, as you showed with RNA, and then thymine for DNA.
Yep. And then I have cytosine and guanine. And again, it's just a shape that I cut out today. And what you can do is you can have a template strand that you give your child. And then you can ask them what the complementary one would be. Oh, and then ask them what the RNA would be. And then with 3 together to get your codon, ask them what the amino acid would be. And so then I have the amino acids all in a thing. And you can work through it like that. And in terms of a puzzle, you could work backwards the more they understand it. Using the codon chart, obviously, no one has that memorized. At least I hope not.
I never did.
You could give them the amino acids and have them work backwards to get to it. The better they get at it. But that, in terms of puzzles, I was thinking about that because my son loves puzzles. And I think that if we approached it more, we do kind of talk about building blocks this way, but I haven't ever seen anybody do it with shapes and color and like pulling it all together. And I feel like you could do this with some pretty young kiddos.
Yeah.
So I'm excited to test this one out, but I just made that today.
I'm impressed. You just creative mind. You just flipped it on. Yeah, that's really great because even the level of seeing, there's 2 shapes that can fit in the same one. When we're talking about, as we started with like the RNA and DNA, like that's different versions of the same information. So I can see that being enjoyed by kids from different ages too. If parents, teachers have a mix of ages with kids, that, if someone's 5 and 10, they could do that together because the 10-year-old may understand a little bit more of like, this is leading to this in terms of the amino acids, whereas maybe the 5-year-old's more looking at the shapes and colors. But they're both being engaged, and, maybe the 10-year-old could teach the 5-year-old a little bit more and everything.
Yeah, and then the older the kids get, you could use this with the kids in high school as well, but have them look more at the shapes of the molecules and maybe have them say what's wrong with this model compared to that? Have them evaluate it and take it a step further. That it's not shaped like a triangle, but it's kind of, and so I think that's a really powerful thing to evaluate a model.
Yeah, and just, visually makes things so much easier because a lot of these concepts with genetics, everything is so abstract. So if there's ways that we can make it concrete in front of someone, I think that just helps so much. And, as we're ending, I just wanted to congratulate you on being a finalist for the 2021 Next Generation Indie Book Award for Children's Juveniles Nonfiction. Tell us about Let's Learn About Chemistry.
Thanks so much. It was really exciting to hear that. I have a fun little medal that I got for it too.
Wow.
It is a book that I wrote, kind of inspired by my son Charlie. He was sorting his toys by color, and I thought, wow, a lot of Gen Chem is actually sorting. These are atoms or molecules. It's a solid, a liquid, or a gas, chemical or physical change, kind of like that. And I was like, I think we could do this. So it is geared toward smaller children. It's It's a board book so that babies can play with it, and it's got pictures of their toys. So they actually do these things with things around their house. So they could actually set up their own grid of 3 things that are similar and 1 that's different. But the way that the illustrator and I worked together on it was that there are multiple right answers. And so the kid could say, these are all red. I could eat all these, or whatever they say, as long as they're providing evidence for their reasoning and it's in the picture. It's cool. Let's move on. You don't have to talk about the science. And the older they get, you start bringing in that second page with the answers for the parents, and you just keep going with it.
I love that. Evidence-based science right there. Well, as we close out, I just wanted to give like a shout out that, you know, you are a science communicator and very active on social media. Any advice for other people that are budding science communicators as you've found success online?
Stick it out. There are ebbs and flows. The algorithm sometimes picks you up and sometimes it doesn't. And it's important to look at it in terms of being an expert versus not. So we can't just walk into social media and think, I'm going to go viral, because it's got this whole wealth of knowledge around it that I'm just assuming. Like, I thought for sure I would be Instagram easy, you know, and it took like a year to get it to a point, and I worked on it every day, and it was like, whew, okay, now I'll work on TikTok. And so now I'm starting Facebook. Like, so I'm getting through these, and I'm starting to pick up tips and tricks that people watch. And so you learn just like you do in school. So there are experts out there, and you can hire them to help you if you need to, if you've got extra cash around. I really recommend it, because there are people who can look at some of your content and say, I don't think that's going to do well. Maybe try it this way. So stick it out, ask for help, and then don't assume you're an expert.
Yes, I think that's well said. I mean, we've had this show for 10 years and we're still growing, and, you know, so it's like not every post is going to be, you know, just exploding. But yeah, I would agree with Dr. Ryan just to stick with it. But thank you so much, Dr. Ryan, for coming on the show and getting everyone pumped and excited for DNA Day and having actual activities to do. That's super easy. I mean, you did it in 8 minutes, as I said at the top of the show. So thanks for just getting us excited about DNA Day and encouraging us to teach kids in our lives about genetics.
Yeah, thanks for letting me share it with everybody. I get to geek out and share my science. I'm in.
I know, that's how I feel about every episode of the show. I'm like, oh, wow, I can just like, people are going to listen to me just nerding out about this. Amazing. But to check out all these fun experiments and Dr. Ryan's book, you can follow her at Let's Learn About Science on all the social media, especially Instagram, as you mentioned. You can also visit her website, letslearnaboutscience.com. I say you make it easy with, you know, all they have to remember is Let's Learn About Science, which is just so simple. But thanks again, Dr. Ryan. Really appreciate you joining us. For more information about today's episode, visit dnapodcast.com, where you can also stream all episodes of the show. We encourage your questions, comments, guest pitches, and ideas. Send them all into info@dnapodcast.com. Search DNA Today on Twitter, Instagram, YouTube, Facebook, so you can connect with us there. And a favor, please rate and review the podcast on Apple, Spotify, or wherever you listen. This truly helps us climb the charts and allow more genetic nerds like yourself to find the show. DNA Today is hosted and produced by myself, Keir Deneen. Our social media lead is Corinne Merlino. Our video lead is Amanda Andrioli. Thanks for listening, and join us next time to discover new advances in the world of genetics.
The genes of you and me, the genes of you and me, are all made of DNA. We're all made of the same chemical DNA. We're all made of DNA.